Okay, so let's begin. Hello everyone, and welcome to this webinar in which we'll be discussing sample size for complex endpoints. And we'll be focusing on three endpoints in particular: Ordinal, Composite, and complex survival analysis endpoints. And for each of these endpoints, we'll hope to give a comprehensive case study. Okay, so my own name is Brian Fox. I am a research statistician here at nQuery for the last almost 4 years, and I'll be your host for this webinar. So just to quickly touch on what the agenda is for today, we'll focus mainly on, as mentioned, three different types of complex endpoints. I'll initially actually just discuss complex endpoints in general, then I'll go into focusing on the ordinal endpoint case, and we'll look at an example of this within the software, and also it's a real life example, and we'll try to discuss some of the issues that one might come across when dealing with this type of endpoint. In a very similar manner, we'll then go on to discuss composite endpoints, and once again look at a case study, although it won't be quite as comprehensive. And then finally, we'll look at complex survival analysis case, or well, in particular, the issue of non-proportional hazards and how that adds complexity to that type of endpoint. And then once again, we'll be looking at a case study for that case. Then finally, I'll aim to wrap things up with a brief discussion and conclusion, highlighting some of the main points to note from this webinar, and maybe giving people an idea of any kind of questions they might want to ask at the end. And I'll leave quite a bit of time at the end of the webinar for any questions that pop up. So feel free to ask away during the course of the webinar. And also I'll give some relevant contact details should anyone wish to send on emails or anything like that. Just to note that I'd probably expect to be well within the hour for this webinar. So there should be plenty of time at the end for questions. Also, actually, if anyone has any issues with connections or any technical issues, just let us know, and hopefully we'll be able to resolve these for you. Okay, so that's probably enough housekeeping. We'll get started. So, as many of you have probably heard us mention before, nQuery is widely regarded as being a cost-effective and easy-to-use trial design platform, which can be an essential tool in optimizing your clinical trial. nQuery is known as the complete trial design platform to make clinical trials faster, less costly, and more successful. nQuery is one of the most widely used sample size and power calculation softwares available and has been in the market for over 20 years. And we're always looking to expand upon the software. For example, in recent years, we added the prediction module, and anyone looking to learn more about that, just let us know. Okay, so in 2021, nQuery is recognized by the leading regulatory agencies, or was recognized as always. And last year, 88% of organizations with FDA-approved clinical trials had used nQuery to some degree for their study designs. So currently more than 50,000 researchers from various parts of the world rely on nQuery to make their trials as efficient and ethical as possible. Okay, so now let's finally get into the statistical part of today's webinar and the main topic, which are complex clinical trial endpoints. And in particular, I suppose, some of the challenges and solutions which are encountered with some of these type of endpoints. So just to mention, I suppose, in case anyone's unsure, endpoints are simply just measurements of what happens to patients within the trial. However, maybe as some of you may not be as aware, in recent years, there does seem to have been an increase in the complexity of endpoints encountered in modern clinical trials. This kind of complexity has generally come from cases where like encapsulating as much information as possible regarding something like the treatment effect is quite difficult to do in just a single endpoint or even multiple endpoints. And so that's where the kind of general complexity comes into play. So some of the main examples of this, which I will be exploring in this webinar, are ordinal, composite, and complex survival endpoints. Just to mention that the three main methods that I'll be discussing for each of these different types of endpoints are proportional odds for the area of ordinal endpoints, the win ratio for composite endpoints, and maximum combination tests for the issue of non-proportional hazards in complex survival endpoints. Okay, and I'll discuss what these are in quite a bit more detail later in the webinar, and I'll also touch on some of the other methods shown here. Okay, so now let's get into the first endpoint that we'll be discussing, which are ordinal endpoints. So it's quite simple, as you can imagine, and I'm sure any of you probably came across this in your first couple days of studying statistics, but basically ordinal data is simply just data that can be ordered into separate categories, or well, it's a data which has an underlying kind of natural structure to its categories. Some good examples of this might be tumor grades, the Likert scale for survey responses, or medical imagery assessment, etc. Whereas something like, nominal categorical data would be something like genders, for example, where there's no underlying natural ordering to the categories. So as you can imagine, this is slightly alien to an area like statistics, where we're used to dealing with quantitative data. So this obviously introduces some issues or complications in terms of how to analyze this type of data or these types of endpoints. Unfortunately, it's often been dealt with quite inadequately, less than optimally, one might say. So very often, the underlying structure is simply either ignored or it's oversimplified, or else approximations are made in order to map it onto existing kind of methods that researchers are more familiar with, rather than actually using an appropriate method for this type of data. So two of the most common methods, I should say, are the Wilcoxon rank sum test, or the proportional odds model. And the Wilcoxon rank sum test or the Mann-Whitney U test, they're basically the same thing. And they're quite simple. I'll discuss that a bit more on the next slide. And yeah, just to mention once again, the proportional odds is what we're interested in here for the purposes of this webinar, and it's what I'll be going into the most detail on. Okay, so as mentioned, some of the most common solutions to dealing with this type of endpoint are simply just to use approximations or simplifications in order to map the ordinal data onto an existing type of data that a researcher might be more familiar with. So this often results in very strong assumptions being made, or similarly, it might end up ignoring a lot of the underlying structure of the data. So I suppose a good example of that might be something like the Likert scale, for example, since I just mentioned that one earlier. So that could technically be mapped onto something like a continuous scale and then analyzed using a t-test. Or also, I suppose you could split it into just, positive and negative responses or something like that, switching into a binary data type. And then you could analyze it using existing kind of proportion related tests. So, the main issues with this is that you're either creating very strong assumptions, in order to map onto a continuous scale, for example, or else you're just throwing away a lot of the underlying information or the underlying data structure. For example, if you are conducting a binomial type analysis. Okay, so the kind of most common, or one of the most common methods to dealing with this type of endpoint is the Mann-Whitney U test. So named just because the underlying test statistic is assumed to have a U distribution. And basically all this does is make comparisons between the different subjects within the trial and assign different values depending on those comparisons, and then summing over those values gives different ranks. So as you can imagine, this is a very flexible method, quite broad in terms of its applicability. However, you are only considering the ranks within this type of analysis. So, you could you could argue that it's sort of ignoring the magnitude of any underlying differences. But once again, it is quite flexible. But this is kind of where the proportional odds model comes into its own, since you're kind of examining the underlying log odds ratio, or odds ratio, I suppose I should say. And this allows for the magnitude of differences through through the use of the odds ratio. And indeed, the reason it's so named, proportional odds model, is just because an assumption is made regarding proportional odds. So just to explain that a bit further, what is the proportional odds assumption? It's quite self-explanatory, but basically you're just assuming that the odds ratio is constant across the different categories of the underlying endpoint. For example, if you're different categories where good, okay, bad, then you'd be assuming that the odds ratio between control and treatment in each of those categories is the same, in case that makes sense. So just kind of explain what the proportional odds model actually is. So here I'm just referring to the proportional odds model, which was proposed by Whitehead in his 1993 paper, which is included at the references at the end of this presentation. It's just based on analyzing the log odds ratio of the proportion of responses in each of the categories under the assumption of proportional odds. And indeed, I suppose from a sample size perspective, at least, this data can actually be analyzed with a z-test, ordinal or ordered logistic regression, ordered logit model, proportional odds model, as we're looking at here, or else indeed the Mann-Whitney test. And indeed, these are all kind of, or sorry, I should say the sample size formula, which I've shown here on the right, which is based on the Whitehead proportional odds model, is actually quite in agreement with any of those methods I've just mentioned. So the reason I'm focusing on the proportional odds model is just that it provides it's a very simple method, and it's very easy to implement. So it's very easy for researchers to adapt to. Just to mention as well before I move on that it does require one small assumption, just worth noting in the back of your mind, which is that it assumes that lower categories imply better outcomes. So by that I mean instead of ordering something in terms of very bad, bad, good, very good, you'd order it in terms of very good, good, bad, very bad. Okay, so hopefully that makes sense. So here we see an example relating to progesterone for severe traumatic brain injury. This is a real trial which completed a couple of years ago, I believe. It was a phase 3 trial, and it actually ended up failing, I believe. But anyway, I suppose that doesn't matter too much. Really, what we're interested in is the type of endpoint which they used, which was based on the the Glasgow overall scale, I believe, or sorry, Glasgow outcome scale. And this was split up into 4 different categories. The first being a good outcome, the next being a moderate disability, the next being severe disability, and then finally they combined both vegetative states and death. So these are 4 ordinal categorical categories. And it's probably worth mentioning as well that these are ordered in the correct manner, so you have the best to worst, rather than worst to best, or least favorable to most favorable. So from a sample size perspective, really what we're interested in is that they found 590 subjects per group. I should mention that they did indeed use the Whitehead proportional odds model in order to determine their sample size, as is mentioned here. However, as we'll see, this is slightly different to the answer you'll get if you use this formula directly. So some approximations, or a bit of or not approximation, sorry, some rounding takes place, but we'll look at that in just a moment. And then, yes, so the other things to note in terms of the calculation we'll be conducting are that they used 1% two-sided significance level. The categories we've covered, they're assuming equal sample size allocation, and they're assuming an odds ratio of 1.5 with 90% power. So now let's take a look at this in nQuery. So in nQuery, one would use this table here in order to conduct this type of analysis. So this is just a very simple table, and this kind of structure goes to show how simple this method actually is to implement. So just to explain for anyone who's not too familiar with nQuery before, basically we have this main table on the left, which is where we'll be entering in all our parameter values that are related to the calculation. And then you'll have a side table just for this particular method. It requires a side table in order to enter in the proportions in the control group for each category. Okay, so I suppose we'll do that now actually before I move on. So as far as I remember, I'll just go back and check. So we have 31%, 21%, 19%, and 29%. So as you can imagine, these all sum to 1. So 0.29, I believe. Oh no, 21 and 0.29 was it? I'll just double check. Perfect. Okay, so there are different proportions. If, for example, you did enter in incorrect proportion, like if you entered in 0.2, then it would rescale all these values. I'm not going to do it here because I don't have to retype them all in, but the software will automatically rescale those values for you. And then once you enter in your odds ratio, which we have as being 1.5, you'll see that the proportions in the treatment group will automatically be calculated for you. And that's done based on this formula here, which is shown in the Whitehead paper in quite a bit more detail, but it is also discussed here. Basically, because of that assumption of proportional odds, you're able to calculate the proportion for each group, given just the odds ratio and the proportion in group 2, or sorry, one of the group proportions. Okay, so let's continue. So very simply, we're just assuming equal allocation, and we have a power of 90%, I believe. And we can see that we get a very, very similar sample size to what the researchers did, which was 590, whereas here we're just getting 586. And usually this is just due to rounding, rounding up in the researcher's case. Okay, so like hopefully I've kind of shown there, hopefully I didn't go through too quickly now, but hopefully I've shown that it is a very simple method to implement. Like this is probably the most complicated formula, and the rest is very simple and easy for not just researchers, but also doctors or clinicians to understand and grasp, since it's quite a tangible way of talking about these types of endpoints. Okay, so I'm just conscious of time, so let's move on to our next endpoint, which are composite endpoints. Okay, so obviously, as you can imagine, composite endpoints are composed or comprised of multiple constituent endpoints. This type of endpoint is actually very common in the areas of like cardiovascular disease, for example. And the example I've shown here shows 3 different endpoints, which would all be included in one composite endpoint, quite commonly, in fact. So cardiovascular death being the most important one, and then non-fatal myocardial infarction being the next in terms of hierarchy, and then non-fatal stroke being the next most important in terms of the hierarchy of these types of endpoints. So I should mention that yes, it's not just having multiple constituent endpoints of equal importance. It's usually referring to constituent endpoints which can be ordered in some way. So sort of similar to the ordinal data structure, if that makes sense. And so as you can imagine, this is a very complex type of data type, or endpoint, I should say. And once again, similar to ordinal data or ordinal endpoints is very often inadequately accounted for or in the analysis. Very often the secondary endpoints are simply ignored from analysis point of view, or from a sample size point of view, especially actually, since very often people are most interested in reducing sample size, so reducing both the study duration and the cost usually, and considering these secondary endpoints can very often do that. So I'll explore that in a bit more detail later when I talk about the win ratio. But just to mention that here, so there are benefits to analyzing these secondary endpoints rather than just ignoring all this information, and that is usually quite expensive to collect. So yes, it's definitely a very important issue, and one which is definitely sort of dealt with inadequately in the opinion of lots of clinicians at the moment. So just to mention that there are existing methods which can deal with this type of data. One of the main ones that people might come across are complex gatekeeping procedures. However, these are very often kind of underutilized, and they actually can be quite difficult to implement in some cases. So that brings to the fore other kind of more simple methods, or maybe not even more simple, just more intuitive methods. So good examples of these would be the win ratio, the win odds ratio, the Finkelstein-Schoenfeld method, and the Boyce's proportion in favor method. So all of these are actually quite simple, or sorry, similar. The Boyce's or Boyce's, forgive my pronunciation of that. The Boyce's proportion in favor of treatment is quite similar to the win ratio, and the win ratio itself, actually I should mention, is just the ratio of wins to losses. However, with the Boyce's proportion in favor method, you're using the difference between the number of wins and the number of losses divided by the total number of comparisons. So that's its test statistic. The Finkelstein-Schoenfeld method conversely examines all pairs of patients and assigns a value of 1, -1, and 0 to wins, losses, and ties respectively. And I'll talk about what wins, losses, and ties mean in just a moment. These values of 1, -1, and 0 are assigned to each comparison. And then the rank for each patient is then the sum of these comparison values. So these three methods, or four methods in fact, are actually very, very similar and can all actually be expressed just by the number of wins and losses if you do it in a certain way. So it's possible to connect the three methods, and this connection is actually what's used in combination with some approximations in order to obtain the sample size formula for the win ratio, which I'll talk about in just a moment. So just to mention that all these methods similarly compare pairs of patient data, which is based on the method proposed by Mann-Whitney, which we touched on earlier. However, the win ratio is just generally the most common, and that's likely due to the fact that for time to event endpoints, it gives an estimate of the treatment benefit, which equates to the reciprocal of the hazard ratio. Okay, so what is the win ratio? I mentioned it briefly earlier, but it's basically just defined as the ratio of wins to losses, and it was first proposed by Pocock et al. in their 2012 paper. So a win implies that a subject from a treatment group gives a more favorable outcome than a subject in the control group. So as you can imagine, then a loss refers to the case where subject from the treatment group gives a response which is worse than that in the control group. And then a tie is the case where the comparison is indeterminate, so where maybe for like ordinary survival, they're both at a similar time or very similar time. And so if there is a tie, then the reason that the win ratio becomes so powerful for composite endpoints is that if there's a tie in the first endpoint, for example, cardiovascular death, as we mentioned earlier, then you proceed to the next endpoint. You try and determine if that comparison results in a win or a loss. And if it doesn't, and it is indeterminate, or it's a tie, then you proceed to the final endpoint, which was, what was it again? It's non-fatal stroke. Then you examine non-fatal stroke between the subject from the treatment and control group, and if that results in a tie, then it is marked down as a tie for that comparison. So this means that you're basically examining all of your endpoints just in a hierarchical manner. So you're ordering your endpoints in some hierarchical manner in terms of priority. So this is very useful, as you can imagine, not just from like a power perspective, but also it means that you can combine lots of different types of endpoints. Like there are other methods that deal with composite endpoints, but very few of them actually allow for the combination of different types of endpoints. Okay, so just to mention that this also gives, this is a bit of a mouthful, but it gives a non-parametrically interpretable effect size, by which I mean that you're not making any strong assumptions, or any assumptions really, regarding the underlying data structure. For example, as we did earlier with the proportional odds model, where we assume proportional odds, that's not an issue in this case. So this is very useful, as you can imagine, and it means that it's quite broad in terms of its application. Although I should mention that maybe it's most commonly encountered in survival type analysis. Okay. Oh, and then quickly, just because we are parents in sample size calculation software, it's probably worth mentioning here that the formulae used for sample size calculation for the win ratio, the ones that we'll be using anyway, are shown here on the right. They're from the paper by Yu and Ganju from 2021, namely the formula on top here. As you can imagine, it's using a normal approximation. And also it makes another assumption, which is probably worth mentioning, which is the idea of no transitivity. So by intransitivity here, I am referring to the case where one pairwise comparison doesn't follow the logic of two other related pairwise comparisons, when you're considering 3 different patients. So this can occur when unequal follow-up times are encountered, where, for example, patient 1 might win in relation to patient 2, and patient 2 might win in relation to patient 3, but then patient 1 might lose or tie against patient 3. So the method here was shown to be moderately robust to intransitivity in that paper by Yu and Ganju. And this paper indeed does go into a lot more detail on that issue for anyone looking to learn a little bit more about it. Okay, so I'll move on to the example. So, my apologies. So this example is pretty simple. It's basically from that paper once again. You'll hear me reference it quite a bit. So that paper, because it's so new, there haven't been very many, or any that I could find anyway, trials that have utilized this this method for their sample size determination. But the paper itself gives very good examples and justification for it. So this particular example is actually a simulation-based example. We can see here the way they kind of set up their simulation, where the primary endpoint is the mortality, and the survival times for this are drawn from exponential distributions with a 1-year mortality rate of 40% in the control arm, and a hazard ratio that ranges between 0.5 and 0.8. However, for the example that we're looking at here, we're going to be assuming that the hazard ratio was 0.7. So the second endpoint in this composite endpoint is recurrent hospitalizations, which are assumed to follow a negative binomial distribution. And each patient's hospitalization times were generated from a Poisson process, where the rate used in this Poisson process was generated from a gamma distribution, The parameters of that gamma distribution were chosen such that the resulting Gammon-Poisson mixture of event times was negatively binomially distributed with the desired mean event rates and dispersion parameters, which kind of affected the number of ties. Okay, so that's probably enough on that. Just from a sample size perspective before we move on, we'll just quickly note that they found a requirement of 250 patients per group, and that resulted in a power of 83.8. So now let's take a look at this in nQuery. We'll be using this table here. So once again, this table is quite simple. There's no side tables or anything like that. All the parameters necessary are encapsulated up here on the top left. So basically, you can imagine these relate to the proportions of wins, losses, and ties that I mentioned previously. And then we can enter in our win ratio, which is just a ratio of wins to losses. So let's do that. So just to double check, our significance level is 0.5, two-sided analysis. The proportion of wins and losses can automatically be calculated if the proportion of ties and the win ratio are entered. So we'll do that here. So I believe it was 1.43, I think. Let's double check. Yes, okay. And we'll assume equal sample size allocation and our power of 83.8, I believe it was. And we can see that our sample size does indeed come out to be 250, which is somewhat impressive given the fact that this is a simulation study. So within the paper, they talk about this, but because the normal approximation was used, the notion of testing the formula, this formula here, against lower sample size values was very important. And so they found that in this case, the simulated power was 83.9, so it's very much in agreement, and that was the case for most of the cases they looked at. I think the maximum difference they found for a case where there's only 100 patients, or the sample size was only 100, was still below 2%. So it just goes to show, like, it's quite a simple method. Once again, like very easy to discuss and to implement. But it is quite powerful and very useful, as you can imagine, for conducting a sample size analysis in a case where very often that mightn't have even been done. You know, it might have just been ignored and some other type of analysis used to determine the sample size. Okay, so yeah, I suppose let's move on. Obviously, once again, just to mention at this point, any questions that come through, feel free to send them on. I know I'm going through this moderately quickly. So yeah, any specific details that I might have missed or anything like that, just let me know. And I'm happy to answer them. Okay. Oh, excuse me. Okay, so the next type of analysis or endpoint that we'll be exploring are complex survival analysis endpoints. And in particular, we'll be looking at the issue of non-proportional hazards. So survival trials or time to event trials are obviously some of the most commonly encountered in the clinical trial landscape, being particularly common in the areas of oncology, cardiology, immune response trials, and so on. In addition, unfortunately, they just tend to require some of the most complex modeling and calculation procedures due to the kind of inherent additional layer of complexity that's associated with these types of endpoints. And in addition, there are, you know, the additional ethical or safety concerns that are usually associated with the subjects in these types of trials. The most common endpoints in this area would be ordinary survival or mortality, progression-free survival, and hospitalizations, for example. Here we will just be focusing on some of the most common numerical or statistical issues likely to be encountered, which add complexity to these types of endpoints. The main examples of these issues are those related to non-proportional hazards, varying accrual patterns, stratification of effects among different cohorts, etc. So there's a lot of research done into this area, or many of these areas, around many of these issues. But some of the more common ones and interesting ones are those of combination tests, restricted mean survival times, and a few others that I'll talk about in just a moment. Okay, so just to focus in on what we're interested in, which are non-proportional hazards. So as you can imagine, both the Cox proportional hazards model and the log rank test rely on the proportional hazards assumption in order for their results to be interpretable. So just for clarity, the proportional hazards assumption just assumes that the hazard ratio between the groups is constant over time. They're, i.e. they're proportional. So as is done in many situations, simply ignoring this case, where it doesn't seem to hold, can lead to very significant issues with regards to the inferences that can be drawn from these tests. And so the added complexity that is associated with this type of endpoint when the assumption doesn't hold is what we'll be focusing on today. So since treatment differences under non-proportional hazards constitute a broad class of alternative hypotheses, finding one test and one estimate of treatment benefit that are constantly or consistently meaningful and provide just good statistical power in multiple situations is quite challenging. So well-known non-proportional well-known non-proportional hazard types which are observed in clinical trials are also shown on the right here. These would include delayed or diminishing treatment effects, or also crossing or hazard effects. And so there are also other issues like responders versus non-responders, frailties, etc. And these issues are most commonly encountered in kind of immunotherapies, I suppose, especially the issue of delayed effects. So there are many different methods which have been proposed to solve these issues. However, as usually seems to be the case, there is a little consensus on a universal best approach. So just to show some of these methods, these are the most common general methods which are used to deal with non-proportional hazards, and also some of the more specific examples of tests which can be used are shown in the right-hand column here. So obviously the log rank or Cox proportional hazards models are often just used somewhat incorrectly, and the average hazard ratio is taken. So that'll probably be one of the most common kind of approximations that's made. A slight improvement upon this would be to use weighted tests, and the majority of, if not all of, the methods proposed just here are using some form of a weighted log rank test as their basis. For the Fleming-Harrington method in particular, it's quite important to note that here, since this is actually the one that we'll be using in our maximum combination test later. Within the example, but yeah, I suppose I won't mention any of the other ones. So I suppose in addition, multiple piecewise models exist, like the delayed effect weighted log rank test, which is great if the survival curves can be split up into separate chunks, which individually satisfy the proportional hazards assumption. Okay, and so the next row, I suppose, is what we're most interested in, which are combination tests. So within these tests, multiple test statistics are considered at the same time in order to handle multiple possibilities for different types of non-proportional hazards when trial data emerges. The most common method encountered in this area is the maximum combination test, which in itself has many variants, but later we'll focus on the one incorporating the Fleming-Harrington type tests into its models. So restricted mean survival time is also another commonly encountered method, but I won't go into that too much more detail here. Let me know if you want me to talk about that at the end. And similar manner, I probably won't go into the reference relative time. And there are many other methods, responder-based trials, which are actually very similar to stratified analysis. There's frailty models, and as we've just covered, win ratio. So you can imagine it's quite a broad method, the win ratio. Okay, so now just because I'm conscious of the time, I'll move on. So just to quickly set the scene and reiterate some of my earlier points, non-proportional hazards is a very common issue in survival analysis and is often dealt with insufficiently. As we've just seen, many different types of non-proportional hazards can occur, and there are a large amount of methods available to combat this issue. Um, so methods like the piecewise weighted log rank tests and restricted mean survival times both require specification of the treatment effect in advance, prior to the study commencing. And while there may be speculation around the nature of the treatment effect, it can often be wrong. So this obviously can further complicates the trial design and treatment effect quantification. So likely adding a fair degree of complexity to the relative endpoint being used. Therefore, combination tests, can be seen as a suitable design and analysis method for time to event data with potential non-proportional hazards that can, describe multiple different alternative hypotheses in a meaningful way, as well as provide competitive power to optimal tests across different scenarios. And hopefully this will make a bit more sense when we look at the example in just a moment. So, just to mention that, like in these cases, or the case where non-proportional hazards doesn't hold, usually a single treatment effect summary isn't really adequate in order to capture the kind of time-dependent nature of the treatment effect or the benefit. And this would imply that we need measures beyond just a simple hazard ratio or restricted mean survival time to quantify and communicate the treatment effect. And so this is where combination tests come in. So the general idea of combination tests is that they are basically just a unified approach to sample size determination for the unweighted and weighted log rank tests under both proportional hazards and non-proportional hazard patterns on the basis of the formulae which are established under local alternative hypotheses. And so they can aim to introduce an efficient algorithm to implement the sample size calculation, usually with strong control of the type 1 error. So the kind of issue that these designs aim to solve is that when the non-proportional hazard pattern is unknown, it is somewhat difficult to pre-specify an efficient weighting scheme in the log rank test. And as one can imagine, the power can be dramatically reduced if an inappropriate weight is employed. So the maximum combination test is one of the most commonly used methods in this area, and is based on assessing multiple combinations of usually Fleming-Harrington-based weighted tests. And in fact, the maximum combination method was a recommendation of a guidance paper produced by a cross-pharma working group on non-proportional hazards. So pretty high praise, as you can imagine. Okay, so basically the maximum combination test works by assessing multiple combinations of weighted tests, all the while controlling the type 1 error. So the main benefit is that it doesn't assume a constant sample size ratio over time, whereas many of the other methods that I touched on earlier do, like for example the Schoenfeld and Friedman methods. So another benefit of it is that the power or sample size can be estimated over a range of choices for the patient accrual distribution, and also for many different baseline hazard distributions, like the piecewise exponential, Weibull, etc. And catering for more of these options is definitely something we'll be looking to add to our software in future releases. So let us know if any distributions in particular are interesting to you. But yeah, I suppose I'll move on. So the power of the maximum combination method is based on the multivariate normal distribution. So the calculation of that is, quite an interesting topic, or difficult topic, I suppose. But I won't get into that here. But the Tang 2021 paper, I believe, which I've referenced at the end of these slides, has a lot of detail, not just on that calculation, but also on this method in general, just for anyone looking to learn a bit more. Okay, so now let's move on to the example. So This is just a very simple example. It's completely hypothetical. Unfortunately, it's difficult to find trials which have implemented this sample size method, but it nonetheless is quite useful to look at this here. So you can imagine this is a moderately more complex example or design than the previous two that we've looked at. So we're going to be assuming that the analysis has been split up into 3 different time periods. So that's where the piecewise nature comes into effect. We're going to be assuming that for the first 3 months, there's no treatment effect, so the hazard ratio is equal to 1. And then for the second and third periods, we're going to be assuming a hazard ratio of 0.75 and 0.6 respectively. From a sample size point of view, I suppose we'll just be looking at these parameters on the right here. We'll be assuming that 4 different tests are used, and I'll explain what these are in just a moment within nQuery. And as mentioned, yeah, we're looking at 3 different periods. The rest of these parameters are quite simple, just similar to what you'd expect from a simple log rank test. So I won't go into these here. Okay, so now let's go into nQuery, and we will be using this table here. So I believe it was a one-sided test. We're looking at 4 different tests. So yes, I'll wait until after actually before I talk about these, but just to mention that I will come back to those. I'm not just skipping over it. So we have 3 different periods. We'll assume our initial accrual proportion is 0. Our accrual period, I believe, was 6. Yes, sorry, 6. And then, whoops, our maximum following period was 9. And our accrual rate is 0, which essentially just means that we're assuming uniform accrual. And then we had an equal sample size. Okay, so and let's enter in our periods, or the end of our periods. So 3, 6, and 9 months. We're going to be assuming that the treatment hazard rate is constant. I think it was 0.077. Just double check that. Yes, okay. And we'll be assuming that for the first time point, there's no treatment effect. And then we can assume, what was it, our hazard ratio of 0.75 and 0.6. And 0.6. And we can see the resulting treatment group hazard rates. And then just to make the example a bit more simple, we're going to assume that there's no exponential dropout. Sorry, no dropout. Okay, so now we can go back and enter in our power, which was 80%. And we can see it does take a little bit of time, but not too long. And we get a somewhat simple, somewhat uninteresting result, since this is a hypothetical example, but it does kind of show just roughly how quick the calculation is done and how easy it is to perform. So now, as promised, I'm just going to quickly focus on the most interesting part of this method, which are the different tests. That's where the combination comes into play. So in nQuery, when you specify the number of tests, there will automatically be certain tests assumed. So there you can have up to a maximum of 4 different tests. If you specify only 1 test, then it's equivalent to doing the log rank test or log rank method. 2 tests assumes 2 different Fleming-Harrington weights. 3 tests assumes both the log rank test and 2 different Fleming-Harrington weights. And then 4 tests, which is what we've specified here. And we can see down here, it specifies 3 different Fleming-Harrington weighting procedures, and then also the log rank tests. So the Fleming-Harrington weights are basically comprised of 2 parameters, denoted by phi and epsilon. The test weights produced when both of these parameters are set to 0 is actually identical to the unweighted log rank test, which in practical terms just means that all events are equally weighted. And then, the case where the first parameter is 1 and the second is 0, produces weights that are more powerful to detect an earlier separation of effects, i.e. when the effect diminishes over time. And then conversely, the 0-1 weighting emphasizes late effect differences, and as a result is suitable for delayed effects. The case of both parameters equaling 1 results in more weight being applied to middle survival differences, which may occur in the presence of both delayed effect and also treatment switching. So just to note actually, before I move on, that there are many other types of weighting tests or test weights that can be implemented. For example, there's the Wilcoxon weight, the Turn-Ware weight, the Pato-Pato weight, which are all more sensitive to earlier survival differences when compared to Fleming-Harrington. And indeed, the Pato-Pato weight is actually asymptotically equivalent to the Fleming-Harrington 1-0 weight. But yeah, I won't go into that in too much detail here. So more complex weighting schemes have also been proposed. If a lag effect is expected, like these two shown at the end here, but many of these cannot actually be pre-specified in advance. However, these two shown here, the threshold lag and the generalized linear lag can be, and so that's why they're included here. Okay, so I'm just quite conscious of time, so I'm going to move on. But yeah, once again, let me know if you'd like me to go into a bit more detail on this during the questions at the end. Okay, so now we'll just finish up with our discussion and conclusions. So just to quickly recap, so complex endpoints are definitely becoming more common in the clinical trial landscape. Some of the more common issues that are encountered in the areas of ordinal endpoints, composite endpoints, and complex survival endpoints are shown here. And the kind of main one for ordinal endpoints, I suppose, was just an inadequate analysis of these types of endpoints by researchers, or indeed just ignoring the underlying structure of those types of endpoints and using some approximations or assumptions in order to map them onto what some researchers might be more familiar with. So one of the main solutions that we discussed for this was the proportional odds model. As we saw, it's quite a simple and easy to implement method, and obviously has a lovely sample size formula, nice and easy. So it's definitely one that's worth considering if you come across this type of endpoint. Then we looked at composite endpoints, which in a similar manner were found to be very poorly catered for in many cases. And one of the main solutions that we came across for this was the win ratio. It's a very modern method and one which definitely has the potential to provide a lot of benefit to researchers. So then finally, we spent quite a bit of time going into a bit of depth on non-proportional hazards and the issues that this causes with complex survival endpoints. And I suppose the main takeaway from that would probably just be that there's greater understanding required around how to deal with non-proportional hazards, knowing the type of non-proportional hazards that you have and what tests are most applicable in those cases. So a very kind of powerful tool in the case where you're kind of unsure about the non-proportional hazard pattern is that of the MaxCombo test. It's quite simple to implement. Hopefully we saw that there in nQuery. And indeed, it's very common, or it's becoming more common, I should say. Okay, so hopefully that wasn't too quick, but just before we finish up, I'll just quickly sum up the next release we have to our software here in nQuery, since a lot of these methods are similar, are applicable to the area of complex endpoints. So we have spent quite a bit of time introducing a new tool to the software, which allows one to conduct a randomization for your treatment, for your subjects in your trial. This includes multiple different types of randomization, including block randomization and complete randomization. And we have different webinars showcasing that tool, should anyone wish to see that a bit closer. In addition, we've added multiple different survival tables, as we've just seen, the maximum combination test, We've added in a non-inferiority version, and we've kind of expanded upon our suite of maximum combination tests. In addition, we've looked at increasing our accelerated failure time suite. We've looked at paired survival, and we've also actually included confidence intervals for viable parameters. And then also, we just saw pop up once again, but the win ratio has also been included, both normal win ratio and also a stratified weighted win ratio, which I didn't have time to cover today, but obviously let us know if you'd like to see that table or see any examples of it. Okay, so then we've also included quite a few proportion tables, including the Mantel-Hansel, both stratified and non-stratified cases, methods for logistic regression, and also, as we saw today, the proportional odds, or the Whitehead proportional odds model has also been included. From an adaptive design point of view, we spent quite a bit of time and effort to include the Litwin's Phase 2 survival designs, and also we have Potvin's two-stage crossover adaptive trial design for equivalence. Then we also have included two tables relating to blinded sample size re-estimation, both for Poisson rates and over-dispersed Poisson rates. And then finally, we've included some Bayesian tables relating to Bayesian average errors and Bayesian factors. And so this release will be coming up in very soon, hopefully in the next couple of months. And so be on the lookout for that if any of these interest you. Okay, so should you wish to test out or play around with any of the examples that I've shown you today, just let us know, or go to statsolves.com/trial, and you'll get access to a free trial of the newest version of the software, or you can contact your account manager if you're an existing customer, and they'll help you get an extended desktop trial or an online trial, for example. Okay. So now I'll just quickly answer some of the questions that came through. Just take a quick look. Okay, so I probably won't have time to get to all of these, but I'll focus on the first two, or the main two, I suppose. So someone asked, during the proportional odds model slide, what does the A represent in the formula, and what also are the P bar I's? Okay, so yeah, I suppose I didn't really discuss it. Sorry, yeah, so just here. So the A just represents the sample size ratio. It's quite simple. It's the sample size ratio between the treatment and control groups. So that's just the notation that Whitehead used in his paper. And also that's just why it's shown that way. So as for the P bar I's, these are actually the proportion of patients which fall into into each of the I categories for the ordinal endpoint. And indeed, the Whitehead paper explains this in a lot more detail if you're interested in learning more about this formula. So another probably interesting point to note just about this formula, in case it's, well, actually, I suppose I probably shouldn't go into too much detail on it here, but yeah, just let me know if you'd like a little bit more detail on that. I'd be happy to go into it. Okay, so the second question that we got was, can you explain what you mean by restricted mean survival times, as I'm unfamiliar with this concept? So of course, yeah. So yeah, I suppose I did. Once again, I kind of skipped over it quite a bit, but restricted mean survival times are just defined as the area under a survival curve at a specified truncation time point. The interpretation of this value is literally just that it's the average time to event over a certain number of time units following this truncation time. Therefore, you know, the difference in restricted mean survival times can actually be interpreted as the additional mean increase in life expectancy up to the truncation time. For example, like 10 months of additional survival on average in the treatment group. And I suppose that, yeah, like the goal with using restricted mean survival times is that they offer a valid measure of between group differences, whose clinical interpretation is a lot easier than that of the hazard ratio. the restricted mean survival time also has a big advantage over the hazard ratio in the sense that it's model free, and so it's robust to non-proportional hazards. But yeah, I suppose I probably don't have time to go into that in any more detail here. Okay, so that concludes today's webinar, I suppose. If anyone wants to get in contact, feel free to do so at info@statsals.com. Obviously, I'll reply to anyone else who got in contact or who sent in questions after this webinar. And also, obviously, thanks thanks to everyone for listening. Hopefully, it wasn't too long, and hope everyone has a nice day. Thank you.